Okay, hello everyone. Welcome back from the break. So we have the last speaker before lunch today, and it is an honor for me to introduce the next speaker, Dr. Tim Nierhaus, a distinguished scientist in the field of structural biology. Dr. Nierhaus began his academic journey at the University of Duisburg-Essen in Germany, where he earned both a BSc and MSc in medical biology. During the second year of his master's studies, he was selected to join the prestigious lab of Dr. Jan Low at the MRC Laboratory of Molecular Biology in Cambridge, UK, supported by the German National Academic Foundation or Student Stiftung. Following this, Dr. Nierhaus was awarded a highly competitive Boehringer Ingelheim Funds PhD fellowship, which allowed him to remain in Dr. Low's lab to pursue his PhD in biological science. His cutting-edge research focused on the in vitro reconstitution of bacterial cell division using advanced techniques such as X-ray crystallography and cryo-electron microscopy. After completing his PhD, Dr. Nierhaus continued to make significant contributions to the field during a 2-year postdoctoral fellowship where he collaborated with Dr. Buzz Baum's group to study archaeal cell division through cryo-EM. In late 2022, Dr. Nierhaus transitioned into industry, joining the Structural and Biophysical Sciences Department at GSK in Stevenage, where he now focuses on the structural characterization of biopharmaceuticals, advancing the development of novel therapies. Please join me in giving a warm welcome to Dr. Tim Nierhaus.
Yes, hello everyone, and thank you for the nice introduction and for inviting me here to speak to you. So yeah, in the next 20 to 30 minutes, I want to tell you a bit about what it is like to be a structural biologist at GSK or in Big Pharma, and from my personal point of view. So just quick summary of who I am in pictures. So I'm originally German, as most of you might be able to tell from my surname. So, but I'm living in London right now, and I've been living in the UK for about 9 years now. And in my free time, one of my hobbies is to learn Mandarin. I have a cat who we also take on hikes, so we like to go hiking and explore nature. And I enjoy food, not very much the cooking, but a lot of the eating. So yeah, so that's about me, summarized in a quick, one slide. So my career, so I started in Germany at the University of Duisburg-Essen, which is in the rural area, so in the west of Germany. And I did medical biology, which is kind of a mix of molecular biology, biochemistry, and also medicine. But then I discovered that I actually really like protein biochemistry and structural biology. So I had a short internship at MPI Dortmund, and that kind of reinforced my beliefs that I want to go into structural biology. So then I was quite lucky that at my university our master course was kind of split. So we had 1 year of lectures and 1 year of only practicals. So I took that opportunity to look around in the UK and apply for positions to basically do my master's thesis there. And I ended up at the MRC LMB in Cambridge, UK, which is a really good structural biology institute. And it was a really great experience to be there, and I ended up in Dr. Jan Löwe's lab working on reconstitution of bacterial cell division. So I liked it there a lot, so I stayed for a PhD, which was another 4 years, during which I worked on bacterial cell division in E. coli, and I learned X-ray crystallography, but also cryo-EM and protein biochemistry, which was kind of the full structural biology package. And then I stayed on a bit as a postdoc, switching some project a bit to work with Dr. Buzz Baum again, which was cell division still, but this time in archaea, which are kind of really funny and weird organisms. So yeah, but I got a bit more experience in cryo-EM and some cryo-electron tomography experience. And then in late 2022, so basically December, I joined GSK or GlaxoSmithKline as a structural biologist in Stevenage. So for those who don't know UK geography a bit, so this is pretty much England. And then what we also have here, so there's biopharma and medtech companies plotted around here. And then if you zoom in, those big centres, so there's London here, then there's Stevenage, there's Cambridge, and there's Oxford. So Stevenage is pretty much halfway between London and Cambridge. And our department is headed by Chung-Hua Chung, and I mainly focus on negative stain EM, X-ray crystallography, and biopharmaceuticals, so antibody-based drugs. So presumably the biggest thing you're thinking about is, if you think about going to industry, is like what kind of is the thought process and so on. So I wanted to share my thought process, which is quite personal and maybe like not what everyone tells you, but so basically what I started out with is to think about a career in academia and the direction where that will go. So I like structural biology a lot, but I didn't want to be like a kind of structural biologist to just do structural biology. So in my lab, we worked like very problem-oriented. So what I liked also was like synthetic biology and like kind of biotechnology things. So I was like thinking about switching more into that field and then trying to remerge it with structural biology again, which is not easy. So in the end, I decided the only place to do that very efficiently would be America. And then I think there's this two-body problem, which Catherine already introduced quite nicely. So my partner, she left academia in the end, but then we had a lot of problems with the post-COVID era, the post-Trump era, both finding a job, visa applications. So that kind of made the US like a kind of difficult thing. And we also have the kind of common language as English only. So that kind of put a big question mark on the US. And then the other thing is what I here refer to as a risk-reward profile of academia. So it's like the security and stability. So what I really wanted to do was presumably very exciting, but would have had like maybe a 5% chance of succeeding to a group leader position. So in that sense, you're like spending a lot of time and you have short-term contracts, you're moving a lot around. So that was one of the things that didn't like particularly want me to stay in academia. So in terms of my drivers, like, so I'm a quite curious person, so I like to have like lots of frequent like intellectual stimulation. I want to like look at lots of things. Which in academia sometimes it's a bit slow. You learn a lot of techniques, but then as a postdoc, you also want to like refine those, but it's really about your output. I absolutely love lab work, so I wouldn't have taken any job into consideration which doesn't give me lab work. And then I like to have projects completed. That said, I might not be the best person to always achieve that, but I really like to get things off my mind because otherwise I can't stop thinking about them. And during my PhD, I actually started a lot of projects, but in the end, like, I might have been better off like focusing on my main one and get that done faster. And then in terms of industry, like considerations, so it's a new challenge and it's a new experience. So in the end, like, I hadn't had any experience what it's like to be in industry. So I was also thinking of it would be good to know, especially since like once you finish a short postdoc, you're still quite fairly early in your career. But it might be good to know what you can do. And as Catherine already said, I think there's definitely opportunity to move back and forth. So knowing the other side at an earlier position might be quite helpful in just like paving out your next 40 years maybe. Another interesting or like good thing I think is fast-paced projects. So instead of spending 3 years on the same thing, you presumably have 6 months to a year or even shorter time frames. It's quite stable, so you have job stability and you have better salary, and it's like more application oriented. So I was already like thinking that I want to be more like involved in technology things. So that's kind of the thought process that in the end led me to go to industry. That said, I also looked at academic labs, but in the end kind of the industry like new challenge and new experience idea was like what drove me to go into industry. And also I talked to lots of other people and colleagues, and in the end they told me the lab work is more or less the same. So that was like one big bonus for me. So I gave it a try and joined GSK. So for those who don't know, so it's a UK company, it's referred to as Big Pharma, so that's about 10, 15 companies that are very, very big. GSK dates back to 1715, and like GlaxoSmithKline as it is was established by a merger of like Glaxo Wellcome and SmithKline Beecham in 2000. And then in like 2022, they like spun out their consumer healthcare business, so that's GSK as it is right now. So it's 70,000 people worldwide. It's a big organization, and that comes with certain set challenges and requirements. They operate in over 75 countries, and they have 37 manufacturing sites. So that scale is really different to what you will be used to in academia, and that means also you have to like be a lot more organized and collaborative. And yeah, so what does GSK do? So GSK has like 4 main therapeutic areas, which are infectious diseases, HIV, respiratory and immunology, and oncology. And within those, there's like a lot of assets. So there's like 71 assets in 2023, and like multiple projects in the pipeline. And then these companies made, like GSK made about 30 billion sales. So just to give you an idea of the dimensions we are operating in, and it's like 6.2 billion R&D investment. And most of you presumably saw the RSV vaccines coming out, so GSK was heavily involved in that. That's like the most recent major development. So if you're interested in R&D in GSK, so these are the R&D sites in GSK located around the globe. So as you can see, it's quite heavy in Europe, and Stevenage in the UK is kind of, being a UK-based company, that's kind of the main R&D center. And then there's a lot of, another big R&D in the US, and in Spain and Italy there are vaccine sites, and then we also have some sites in the Asian area. So the team I'm in is called Structural and Biophysical Sciences. So it's a group of about 30 people, headed by Chung Hua, who is also like a structural biologist and originally an NMR spectroscopist. And yeah, we're quite a diverse team, and like a bit uncommon for a big pharma. Almost all of us have PhDs. So in industry, you find a lot more people without a PhD, but in structural biology, because training is quite intense, we tend to be like very specialized workforce. So what we do is we use like structural biology and like biophysics to integrate atomic insights into a mechanism of action and pharmacology. So we have like mass spectrometry and like biophysics and structural biology, X-ray crystallography, electron microscopy, and we characterize drugs and like drug target complexes and also antibodies, and we try to understand their properties, but also their mechanism of action, which is the most important thing we try to contribute. So we basically want to know how the drug works, how the pharmacology works, and that's where we bring in our expertise, and that is to deliver differentiated medicines. So like with the genome area, basically lots of the targets are out there. What you need to know is like, why is your drug better and what is special about it? And structural biology can be really helpful in understanding that. And if you want to know a bit more, so Chumwa gave a talk when she was elected as a fellow of the Medical Science Academy. And you can go on YouTube and look at that if you want to find out more. So about the drug discovery process, so where are we? So in general, drug discovery, so you first spend time on identifying useful targets. That's kind of proteins that are like, or like other molecules that are involved specifically in disease and have like good off-target profiles, so you don't want to hit other on-target things. And then what you do is you basically screen and discover potential drugs, and then you optimize them, and then you go into like in vivo studies and development, and eventually you register that. So that whole block here is where we are mainly involved. So it's like the early drug discovery. It's more like the drug optimization and characterization. And here it's also very important to have the molecular mode of action ideally already. And as you can see, so from those timelines for antibody project there, you can presumably assume the left number. So an industry project from having a target to basically going into development takes about 1.5 to 2 years. So you also need to work to those timelines. And then in terms of equipment and lab, it pretty much looks like a structural biology lab at the Max Planck or any other research institute. So we have 2 glaciers in-house. We have access to 2 cryoses via the Cambridge Pharmaceutical Cryo-EM Consortium. So there's 5 pharma and biotech companies that together with Thermo Fisher actually run these microscopes and use them. We have X-ray in-house generator, and we have weekly to fortnightly synchrotron access. And also we have state-of-the-art mass spectrometry and biophysics. And our group is actually quite heavy on mass spectrometry and HDX. And we have capacity for about 500 structures a year. So that's a lot more than what you would do in your PhD, which is presumably 5. So what am I doing? So I'm still working in the lab, which I also wanted. Maybe not as much as I initially anticipated, but I kind of come to terms with it. So I mainly do negative stain characterization of antigen and antibody complexes. I do X-ray crystallography of those complexes, and I do a bit of biochemistry, generating farbs and purifying them, and I do a lot of data processing and analysis as well. So outside the lab, sometimes it's up to 70% of the week. That is because I have a quite special role, so I'm not only a structural biologist, lab scientist, but I also kind of have a role in our department liaising with the biopharm discovery department, which is another 100 people. So what that involves is basically keeping track on their projects and answering their questions around structural biology and molecular mode of action. And that's actually quite interesting because you get a lot of science there. So whereas this part might become a bit repetitive after a while, here you see really a lot of projects. And that's kind of a pre-stage or similar thing, so it's called project management. So there you would just drive the whole project from target discovery all the way to development. So those jobs can be very interesting, but they involve a lot of coordination and matrix working. So I also supervise an industrial PhD student and industrial placement student, so that's a 1-year work internship. That takes quite a bit of time. There's a lot of meetings, so if you join industry, you will presumably be shocked about how many meetings you can be pulled into. So you need to be a bit specific, but they are essential because if you work around 70,000 people worldwide, you need to talk a lot and coordinate things. So I'm also spending a lot of time coordinating the work in our department and the biopharm discovery area. And I do a lot of summary and presentation of results, but also interpret them and try to distill the structural biology information for non-experts. And then there's always process optimization and standardization. So the one question in industry is always, can you do it with less material and can you do it faster? And that really should be on your mindset. And I think I'm quite organized, so I like doing these things. So yeah, what are some differences between working in academia and in big pharma? So I think Catherine gave you a really comprehensive list, so this is maybe more some bullet points that come to mind. So it's a bit exaggerated, but in academia you basically solve one or a few novel structures in 3 years. So in industry you establish a system and then you solve hundreds of structures with X-ray crystallography or still tens with EM. So those ones are not different proteins all the time, but different drugs. So what you basically want to find is the most promising drug candidate out of hundreds or thousands. So you really want to focus down on doing things efficiently and quickly. Whereas in academia you still want novel insights, but here it's really just find the one that looks promising and differentiated from others. In academia, especially if you're doing your PhD and maybe your early postdoc and in structural biology, training can be quite long. You're trying to become an expert, so in industry they expect you to be the expert, so you will just run things. And in academia, people are like very focused on developing technical expertise. So in industry, I think you rather want to develop soft skills, but you want to keep up with your technical expertise. And then, so what I say here is like work alone, be the process. So my lab was especially like people were working on their own projects. But in a PhD, you're most likely working like by yourself or maybe with 2 or 3 other people. Whereas like we cross-collaborate with 30 people. And what's really important is like teamwork and communication. So you might not be the person purifying the protein, you might not be even the person collecting the data, you might just be the one building the model and interpreting it. So you need to know what happened before, and you constantly need to update people. And then you have a more discovery-driven project direction. So my PhD project started out to, it was supposed to be cryo-electron tomography and fluorescence microscopy. In the end, I did X-ray crystallography, because we discovered something interesting and solved the structure of it. But in industry, you have very clear objectives, and you usually have quarterly progress tracking against those. So you want to make, you want to be focused and make sure you do what's important. But that can also be a real good thing if you like to really have a clear-cut plan of what you should do. And then, yes, salaries and personal benefits are more comprehensive, and work hours are a bit shorter. That said, I still sometimes work longer hours and like on weekends, but it's not regular, and it's not as much as in my PhD. So so what do I think like people in industry hiring look for? So they look for a technical expert with deep expertise and of cutting edge technology, and they want you to be familiar with like processes, so like data analysis and everything. They want you to be timeline-oriented and process-oriented and organized, and you absolutely need to be a good communicator and be able to work in a team. Because otherwise, in big pharma with like 70,000 people, or like even for me, I presumably get like emails from 50 people in a month. So you really need to be on top of that and communicate. So for your interview, what I really think you should focus on is what you have done. So from other interviews I saw, so I think people in academia like to use this we. So what industry wants to look for is what you actually have done and what you actually know and are an expert in. So try to tailor your talk a bit onto that. And then demonstrate your soft skills, especially communication, organization, and delivery. So if you have something that you have driven forward, maybe your supervisor wasn't super supportive of in the beginning, but you like thought it's a good idea, that's like prime examples to use. Be prepared to understand the drug discovery process and expectations. And like, there's a lot of acronyms in industry, so reading up on those is quite helpful. And then for me, what was quite important is to prepare for HR interview questions. So those are like very kind of standardized questions, but they're a bit like strange to me at least. So people would ask you like, tell me about the time you succeeded, or you like solved a communication issue and so on. And I wouldn't have been prepared to those, for those except for like preparing actually. So there's like lots of resources on LinkedIn and other similar platforms. So be prepared for those. And then tailor your science talk. So don't have an interview where you spend like 10 minutes, if it's a 20-minute interview, don't spend 10 minutes on the background. Rather give like a succinct background and focus on examples where you actually have done something and like delivered something and show your expertise. So I think that's quite helpful for you to narrow it down, and also I think that's what the people in industry want to hear. So yeah, if you start in industry and get a job, so what I think are the things you should focus on starting off. So I've been there for about 2 years, and I think it's going fairly well. But I can already tell you there's like a lot of things that I would have maybe even paid more attention to if I knew earlier. So you should definitely prioritize delivery and manage your time well. So it's very easy to get distracted because there's like lots and lots of people that might ask you questions. But you should like focus on like your main goals, your main objective, which is like delivery. These companies, big pharmas, are very complicated. So make sure you spend some time to understand them. And then in terms of like visibility and networking, so you're one of 70,000 people, so make sure your work is seen. Show it to others, like talk to others and develop your soft skills, just so that others notice you. Otherwise, nobody might know who the face behind the data is. And also always look at process optimization opportunities. And I think if you want to like advance quite quickly in your career, what's very good is like try to become a people leader. So get responsibility for managing people, that's very well seen. Look at portfolio facing roles, so like general roles that look at all the drugs in the pipeline, and work in project teams or matrix roles, so interdepartmental roles, and matrix working is really important, and that's essentially what all the like senior people in these big companies are doing. Yeah, and just like the last few minutes, I just wanted to spend on telling you a bit about like what I think might be some difference between living in the UK compared to Germany. So I never worked really in Germany, but I've been living in the UK quite a bit, and I still have a lot of friends in Germany, obviously. So the UK is more international. Presumably one of the best things is you can go grocery shopping on Sundays. So when I come back, I'm still confused that things are closed on Sundays. The daily life is more digital, so like cashless pay, deliveries and appointments. Like in Germany, they still have way too much paper, in my opinion. But cost of living, and especially childcare and education, are higher. There's free healthcare in the UK, but that comes with, yeah, it's not always the best compared to the German system in certain aspects. But if you like have a work visa, you also need to pay for it. In terms of working, it's greater flexibility in switching career trajectories. So my partner, she was trained a scientist, now she's a banker, which happens all the time. So, so I think in Germany people are still a bit more like fixed on you do biochemistry, you become a biochemist. It's a bit late riser friendly, so UK is usually 9 to 5, not 8 to 4 or so. Also at GSK we have core working hours from 10 to 4, so you can shift your day quite late. There are not that many probation periods, and they are also usually shorter, which I think, yeah, was something I wasn't really aware of. The CVs in the UK look quite different, so spend some time to look at them, especially if you're like used to German CVs. Salaries in life science in the UK are not the most amazing, but yeah. And post-Brexit, Europeans also need the work visa. But to sum that up, it's pretty much the same. So I don't feel much difference like living there or here, so yeah. Yeah, and if you're interested, like, please check out. Our group is hiring very soon, so yeah, thanks for your attention, and I'm happy to take questions.